Welcome to QUT's Mackay Renewable Biocommodities pilot plant, where we're going to be looking at a process for converting sugarcane bagasse into biofuel ethanol. Well, here we are in the field. This is the sugarcane growing in the field. What happens is this gets harvested and it gets transferred over to the sugar mill, which is right behind our pilot plant here. The cane gets crushed and the juice comes out and we make sugar from that. But what's left over is the bagasse material. It's the fibrous material. It's the thing that's left over. It's a byproduct. This normally gets burnt and generates electricity for the plant and for the town. But what we want to do is convert it to ethanol biofuel. And this happens right here in our plant. So let's go and have a look at actually how the process works. So here we are on the top floor of the pilot plant. If we look around us, we can see things like the chemical storage tanks. There's a spray dryer over here. And this is where the bagasse enters the process.
So what is bagasse composed of?
Well, this is the bagasse here. It's a fibrous material. It's actually made of different polymers. There's lignin, there's cellulose, and there's hemicellulose. And the process we're looking at is breaking down that cellulose into glucose so we can ferment it into ethanol.
So how does the process work?
Well, to start off with, the bagasse comes up this conveyor here, and then it drops into these hoppers, and then moves into the horizontal reactor down the bottom where it gets mixed with either acid or base, heated up, and then stirred by this piston coming in and out.
Can we see it working?
Yeah, sure. Yeah, we can switch it on. Hank, can you start it up? So you see it coming up here, dropping in, coming through the system and then dropping down into the horizontal reactor. So if we head back this way, we can see it from the bottom as well. So here we have the reactor at the bottom. You see the piston moving in and out, and that's mixing the contents of the reactor. So in this process that we've run today, we've actually used alkali or basic conditions, which means in the liquid fraction, we actually get out the lignin stream. So that this liquid lignin stream, it flows out of the reactor we saw before. It comes down this pipe here and flows into this holding tank.
So can we have a look at the lignin in the soluble form?
Yep, sure. So here it is in this vessel here. We can see it coming out. So this is it here.
So what about the cellulose?
Yeah, so the rest of that fibre, that's going to be the part of the process we're going to see next upstairs. Here we are now on the second floor of the pilot plant to see what happened to the fibre. So you can see up here we've got the horizontal reactor that we saw right at the beginning of the process, and the fibrous material has dropped down into this vertical steam explosion reactor.
What happens in the reactor?
So in this reactor, the fibre gets under a really, really high pressure, and that pressure is suddenly released, which explodes all the fibres apart, further breaking it down. And the whole thing is operated from the control room here. So let's go downstairs and see the process happen. Here we are now at the bottom of the steam explosion reactor, and let's see what actually happens. So let's go. We better put in earplugs. It could be quite loud.
Step back.
All right, so let's step back as well.
Oh, that was loud.
All right, well, let's have a look inside.
Wow, that looks different to how it was at the beginning.
Yeah, it's really changed quite a lot. You see it's got a lot darker and it's broken down compared to what it was to start with.
Yep.
So this is now ready to have the enzyme added to break down that cellulose to glucose. So let's go and have a look at that next step. Here we are at the enzyme reactor, and the pre-treated sugarcane bagasse fibre that we saw before has been loaded into this reactor, and then Heng here is now adding the enzyme to start the reaction.
How long does the enzyme take to break down the cellulose?
So it takes about 1 to 2 days to break that cellulose down to the glucose that we need. So it's stirred, it's kept at an ideal temperature for the reaction to occur. And what we end up with at the end after those 1 to 2 days is this glucose-rich syrup that the yeast can grow on and make ethanol. So let's go and have a look at the fermentation. So now we're in the fermentation facility.
Where do you make the ethanol?
So we're going to make the ethanol in the 1,000-litre fermenter here, but to do that we need to make enough yeast.
Where are the yeast?
So we've made the yeast in a starter culture in the lab, and it's now in this bottle here. So we're going to transfer this to a 10-litre fermenter and then a 100-litre fermenter, and then that'll go into the 1,000-litre fermenter along with that glucose solution that we made using the enzyme before, and that'll make us the ethanol from our bagasse. So let's get it started. Here we are now at the bottom of the 1,000-litre fermenter. The fermentation's finished, we've made our cells and we've made our ethanol.
What happens next?
What we're going to do is get the cells out, so we use a continuous centrifuge to do that. That separates the cells out from the broth which contains the ethanol. So that broth is then distilled and we end up with this pure ethanol product here. And that's the end of the process. That was the process for converting sugarcane bagasse into ethanol biofuel. And really what we use this facility for is to do the research around how to make that process as efficient and cost-effective as possible.
So, apart from ethanol, is there anything else that you can do here at the plant?
Yes, we do lots of research here. It's a really flexible facility. So we don't just look at bagasse, we look at other fibrous waste like wood chips or other types of material. We also use a fermentation to make lots of different products as well. We make oils, we make chemicals, we also make even things like animal feed supplements, like enzymes and probiotics. So I hope you found that tour interesting and learnt something today. It's been great to have you here.
I certainly did. It's been great. Thank you.